Today we're here and we're back with another MCAT Max walkthrough. And today we're going to be doing a passage from the bio/biochemistry section of the MCAT about enzymes. And this is a pretty tough passage. And we're using some Jack Westin questions. So let's dive into it and see how it is. So the passage here it tells us about a yeast, Saccharomyces cerevisiae. And it tells us that it peaks its expression of the CDC8 gene, okay, at the G1S transition during the cell cycle. So what is the G1S transition during the cell cycle? Remember the S phase of the cell cycle, the S phase, that is when we replicate the DNA. So for the yeast to peak its expression of the CDC8 gene, that must mean the CDC8 protein. Remember, genes code for proteins. So we go from genes to mRNA to proteins. So for it to peak its expression of the CDC8 gene during the S phase of the cell cycle, that must mean the CDC8 protein helps out with DNA replication. Okay, so keep that in mind. And then it tells us that CDC8 coordinates with other DNA synthesis genes. So other DNA synthesis genes such as CDC21. And CDC21, the protein it codes for is Thymodolate Synthase. And it tells us what Thymodolate Synthase does. So it basically what you need to know is it converts the deoxyuridine monophosphate to deoxythymidine monophosphate. So what is that? So you know DNA is made up of nucleic acids. So a DNA polymer is going to be made up of these nucleic acids. So you're going to have a ribose sugar. Remember, this is what makes up DNA. It's going to be attached to a nitrogenous base. So I'm going to write base here. And then at the 5' end, you're going to have a phosphate group. So So this is a deoxyuridine monophosphate, or basically a deoxynucleotide. This would be not a deoxy. This would be a ribonucleotide phosphate, because it has the OH group here. Remember, DNA is gonna let's see. Let me erase here. DNA. So okay, this right here is a DNMP. DNMP. So a DNMP is a form of a nucleotide. So remember, DNA is made up of nucleotides. So this thymodolate synthase converts the deoxy nucleotide monophosphate, in this case the uridine one, to the thymidine one. So deoxyuridine monophosphate to deoxythymidine monophosphate. So remember, you have your nucleotide, which is a pentose sugar. So 1, 2, 3, 4, 5. So we have a 5-carbon sugar attached to a base. It can be U or T. Remember, this enzyme is going to convert the U to the T. So your base is going to change, but you're still going to have one phosphate group attached here, okay? Okay, let's move on to the next paragraph. It says the CDCA gene codes for an enzyme, which phosphorylates the monophosphate nucleotides. Remember, this is our monophosphate nucleotide, to diphosphate nucleotides. So NMP to NDP, and it requires ATP as its phosphate source, and it needs the the magnesium as a cofactor. So that enzyme needs the magnesium on it for it to function. So that's called a cofactor. And so it says the enzyme is TMPK. So that is the enzyme that CDCA codes for, and it phosphorylates the monophosphate residues to a diphosphate residue. So a diphosphate residue, like it's in the name, is going to have 2 phosphates on the nucleotide. Okay, so the TMPK is going to phosphorylate. So that stands for thymidine monophosphate kinase. And so it converts the monophosphate to the diphosphate. So 1 phosphate to 2 phosphates. So this is 2 phosphates. So this is thymidine deoxythymidine diphosphate. So this is TDP. All right. And so although CDCA expression is known to result in the production of the nucleotide diphosphates, remember this is a nucleotide diphosphate, we need nucleotide triphosphates, so we would need 3 phosphate groups for DNA replication. So we need NTPs are required for the template synthesis of DNA in vivo. So in vivo means in a living organism. And so Assay, the yeast must endogenously produce. So it must itself produce these deoxynucleotide triphosphates, these DNTPs. Whereas the CDCA gene codes for TMPK, which is known to make the diphosphates, but we don't know if it can make triphosphates. So the experiment later on in the passage is just going to kind of look at that. So we're trying to figure out whether this CDCA protein, the TMPK, can also have some TDPK activity. Can it convert the diphosphates to triphosphates? Because we know it can convert the monophosphates to diphosphates. We want to see if it can make triphosphates from diphosphates. Okay, let's move on. So because we know we need to make the deoxynucleotide triphosphates, we want to see how CDCA affects the DNA replication. Because remember, we need the nucleotide triphosphates. So what it says is they made some CDCA protein. So they made a recombinant proteins. They purified the protein. So we have the CDCA protein. Okay, that's what we're using. And it looked at their activities in vitro, so within cells. And so they gave different doses of the CDCA enzyme. And they looked at their activities, okay? And that's what figure 1 is. So let's kind of look at figure 1. So figure 1, it gives us enzyme activities on the y-axis, and it gives us the amount of enzyme. So this is the amount of the CDCA protein. And this is its activity. So here we have the color scale. So DTDP is the gray, light gray, whereas DTTP is the dark gray. So we see here that up to about 1,000, the primary activity of the CDCA enzyme is making DP. Sorry, that's kind of like hard to say, but the deoxythymidine diphosphate. So it's going from I'll use the text function. So it's going from the DTMP to the DTDP. Okay. Whereas if you look here after 1,000, we have some TDP TDPK activity. So this is TMPK activity, right? So it's TMPK activity because we're phosphorylating a monophosphate, right? We're phosphorylating the DTMP to DTDP, so mono to di. What we want to see is if the CDC8 enzyme can make the DTTP, because that is our triphosphate. Remember, we need the triphosphates for DNA replication, so that's what we're looking at. So you want to see if it can go from DTDP to DTTP. So this would be TDPK activity, right? Because we're phosphorylating. This stands for thymidine diphosphate kinase. So we're phosphorylating the DTDP to DTTP. Okay. So what we can see here is around 1,000 nanograms of CDC8. We start to have some TDPK activity. We start making some DTTP. Whereas below 1,000, like for example 3 nanograms here, we mostly have TMPK activity, right? We're mostly just making DTDP. We're mostly making the diphosphate around here. Whereas here, we start making the triphosphate. So we start to get some TDPK activity around 1,000 nanograms. So let's go on. Let's move on down here. So we have a substrate specificity assay, okay? So basically what they did is they gave you some different substrates right here. A uridine monophosphate, deoxyadenosine monophosphate, deoxythymidine monophosphate. And they added the CDC8 protein to it. So they wanted to see what the CDC8 protein could create. So this is going to be our deoxynucleotide diphosphate. This is our deoxynucleotide monophosphate. And this is going to be our deoxynucleotide triphosphate. So remember, we didn't know whether CDC8 could make the triphosphates. We knew it could make the diphosphates, but we did not know whether it can make the triphosphates. So we knew for the DNA replication to happen, the yeast had to make triphosphates. We didn't know whether CDC8 could do that. So the fact that these triphosphates exist right here, that shows that CDC8 has some TDPK activity. So it can phosphorylate the diphosphates, and we already knew it could phosphorylate the monophosphates, right? Okay. And it's called a substrate specificity assay because for the DAMP, the deoxyadenosine monophosphate, remember guys that a nucleotide monophosphate is just a general term for any nucleotide with a phosphate attached to it. So here, this is a deoxyadenosine with one phosphate attached to it. This is a deoxythymidine with one phosphate attached to it. So that is when I say nucleotide monophosphate, nucleotide triphosphate, those are broader terms for this stuff. So a nucleotide triphosphate is a broader term for a deoxyadenosine triphosphate, right? So it's just a specific form of nucleotide triphosphate. Okay? And remember, so nucleotide triphosphate, I'll just draw it out for you guys. It's going to look like a ribose sugar attached to a base. And we're going to have 3 phosphate groups here. So to be Okay, guys, so that is nucleotide triphosphate, and it's this little dot right here. So now that we've gone through the passage, let's look at the questions. And remember, this is a substrate specificity assay, so the fact that there is no nucleotide diphosphate or triphosphate for the DAMP means that it couldn't phosphorylate this monophosphate to make a diphosphate, or phosphorylate the diphosphate to make a triphosphate. So I know this is a hard passage, guys, so feel free to like pause anytime, think through it, and reach out to us if you have any questions. So it says an additional experiment was performed using a CDC8 mutant, which is known to lack TMPK activity at high temperatures. They conduct a substrate phosphorylation assay, but only add UMP, ATP, and Mg2+. Which of the following TLC plates most likely depicts the result of this experiment? Okay, so we know for a fact that CDC8 is going to have TMPK activity. It's going to make these diphosphates. Remember, diphosphates are the second dot right here. So only at high temperatures is our CDCA mutant not going to work. So only at high temperatures. At normal temperatures, our mutant is still going to work normally. So the figure should look just like the normal wild type. It should be the same for the normal mutant and normal wild type. So let's go through and look at which options match that. Okay. So off the bat, we can eliminate A. Oh, no, we can't. We can eliminate B and we can eliminate D because we know our normal mutant and normal wild type are the exact same because only at high temperatures do the mutant and the wild type differ. So we know that it either has to be A or C. Okay. So if it lacks TMPK activity, remember TMPK is monophosphate kinase. If you can't phosphorylate the monophosphates, you can't make diphosphates. So we should have no dot in the second place. So we don't have a dot here. That's good. We don't have a dot here. Okay. So what separates A and C? A does not have any triphosphate. C does have triphosphate. Well, C has to be wrong because if we don't have any diphosphates, we can't make any triphosphates, right? We can't, if there's no diphosphates to phosphorylate, how do we get a triphosphate? A triphosphate comes from phosphorylating a diphosphate. So if you have no diphosphate, you cannot have any triphosphate. So A is the correct answer. And I'm just gonna show you guys nucleotide triphosphate just so you guys can see what that's about. Get a better visual image. So this is a nucleotide triphosphate. Remember, we have 3 phosphate groups attached to a ribose sugar, 5-carbon sugar, with a base attached. So this one right here would be adenine. So this would be deoxyadenosine monophosphate. So remember, we need these triphosphates for DNA replication. But in the yeast, we start off first with just a monophosphate. Then we go mono to di, and then we go di to tri. We didn't know if our CDC8 could make the di. No, we did know that our CDC8 could make the di. We didn't know if it could make the tri. And based on this graph, based on the figure here, we know that it can make some tri. So it does have diphosphate kinase activity. So let's move on to the next question. Given that 20 picomoles per minute is the minimum amount of enzyme activity to produce a sufficient concentration of nucleic acid monomers in vivo, what must be true about S. cerevisiae? So nucleic acid monomers, these are going to be our nucleotides. So that's just another word for nucleotide. So remember, we need the nucleotide triphosphates for us to do DNA replication. So it's saying that we need 20 picomoles per minute of production of the DTTP. So we need 20 picomoles of DTTP produced per minute for us to have a sufficient concentration of nucleic acid monomers. Because remember, we need DNTPs. We need DTTP to do DNA replication. And it tells us here that we need 20 picomoles per minute of the nucleotide triphosphate for us to be good. So when do we have 20 picomoles per minute of DTTP being made? That is around here at 1,000 enzyme nanograms. So when we have 1,000 enzyme nanograms, then replication can occur because we have a 20 picomoles per minute. So the enzyme has an activity of 20 picomoles per minute. So it's producing 20 picomoles of DTTP per minute. So that is when DNA replication can occur. So replication can occur when amounts of CDC8 are above 1,000 nanograms, because when it's above 1,000 nanograms, we have 20 picomoles per minute or more of DTTP being made, because we need the DTTPs for DNA synthesis. So let's look at the other options. It says replication can occur when amounts of CDC8 are above 3 nanograms. So when they're above 3 nanograms, you can see here we have above around 20 picomoles per minute of DTDP being made. We need the DTTP, not the DTDP. So that's wrong. TMPK activity is only present when amounts of CDCA above 3 nanograms are present. That is wrong because TMPK produces TDDP. So we have DTDP, the light gray, being produced below 3. So that is wrong. And it says, the last one says TMPK activity is only present when amounts of CDC8 are above 1,000 nanograms. That's wrong because below 1,000 nanograms, we still have a ton of light gray. We still have a ton of DTDP being produced. Remember, TMPK produces DTDP, the light gray. I know it's a lot. It's pretty confusing, guys, but if you think through it, watch the video and just let us know if you have any questions. That'd be great. Okay, let's move on to the next question. So this is a hard passage. So CDC21 encodes a protein that has what kind of enzyme activity? So where does it talk about CDC21? It talks about CDC21 right here. It says it encodes a Thymodolate Synthase. So a synthase, guys, you just have to know this is a subclass of ligase. So ligase is our correct answer, right? And so ligase forms new bonds, and the synthase is a form of a ligase. So you just have to know that synthase is a form of a ligase. So that's how we answer that question. What type of enzyme acts against the activity TMP kinase? So remember, TMP kinase, it's adding a phosphate group to the monophosphate. So MP stands for monophosphate to make a diphosphate. So an opposite activity would be something that removes a phosphate, and that is the phosphatase. So that's going to be the correct answer there. Because phosphatase removes an enzyme, whereas a kinase adds a phosphate. So phosphatase is going to be the opposite. And it tells us here, the last question, in the context of the TMPK catalyzed reaction, Mg magnesium ion functions as a what kind of molecule? So magnesium is required. Magnesium is required for the TMP kinase to work. So when you need a metal ion on the enzyme for the enzyme to work, this is called a cofactor. So whenever a metal ion is needed for the enzyme to work, that is called a cofactor. You need to know that fact. A prosthetic group is when you have something besides a metal ion. For example, you might know FAD. When FAD needs to be, when a non-protein group needs to be on the enzyme at all times for the enzyme to work, that's called a prosthetic group. Because we have a metal ion here, magnesium is a metal, that is called a cofactor. It's not a prosthetic group. Co-substrate is something that attaches and detaches from the enzyme, so it doesn't need to be on the enzyme at all times. Right? And coenzyme can include coenzymes can include prosthetic groups or co-substrates. So that's a broader term for each of these. But the correct answer is cofactor. All right. And that is the end of the passage, guys. Let's make sure we got everything correct. Sorry, guys. I had a technical issue there, so I had to cut. But as you can see, we got every question correct on this. So great job, and hope to see you another day.